it doesn't. Okay, we go with this. So, you know, vaccines are in particular for infectious diseases, and for infectious diseases of viruses, for instance, that are produce acute disease, you need antibody responses, and antibodies are in most vaccines are directed against proteins, or including sometimes epitopes that contain glycans, and I don't have to go into the details of how complex is the initiation of the immune response. So you need certainly antigens, and you need a large number of different subpopulations, and Federica was actually showing the complexity of the dendritic cell environment. And you need these cells to activate T cells, which in turn actually are essential also to induce activation and proliferation of B cells in order to produce the differentiated plasma cells. And then you get your antibody expressed and produced in the serum. And in order to get all this, you need to have, obviously, you need antigen. But the antigen needs to be in the right amounts, in the right availability, but also in the right conformation in many cases. And this is particularly the case in which we go into the genetic immunization, so the so-called genetic vaccines or DNA vaccines. I think the term genetics is obviously more correct, since you can have vaccines which are also genetic based on RNA and not on DNA. But the most common ones are the ones on DNA, and these are the ones that we are also implementing. So what this is particular on genetic vaccines is that you do not give the antigen itself. What you provide is the gene that encodes the antigen. So you need a transcription unit that actually gets internalized by cells of the host, and those cells are the ones that produce the antigen. And this is it's close to what happens when you get an infection, for instance, with a virus. So you do not make antibodies against the virus that actually comes to infect you. You make antibodies against the protein produced by the virus, by the cells infected by the virus. So it is the product of the infected cell what actually stimulates the immune system. And from that point of view, the genetic immunization is a close surrogate of actually the infection. Of course, you need other elements. Many times, and most of the times, what helps to obtain a good immune response is inflammation. Now, as I said, you get expression of the antigen is produced by cells of the host. And from that point of view, the antigen produced has the same post-translational modification as the one you get when you get infection. And this is true. I'm concentrating mainly on viruses, and you will see why. I will be describing one example that we are working on. And one element that is extremely important in all the strategies of genetic immunization is the production of the antigen, so the availability, the secretion of the antigen from the cells. And this is an aspect which is very important and has been largely neglected. You see many publications of people trying to get DNA vaccine in which you get the gene, they drop it there into an animal, and they look whether you have this type of response, or you have T cells or B cells or whatever. And nobody asks the question, how much do I have really antigen, do I produce? In many cases, the failures of the very low levels are due to the fact that the antigen is not in the right amounts, or even in the correct conformation, and we will go into that in a minute. So our interest was actually to try to implement the development of a genetic vaccine for a disease which is a dengue virus. And fortunately, Alessandro Marcello already presented the many characteristics of a flavivirus. Dengue is a flavivirus, and here you have a nice 3D reconstruction of the virus. These are the numbers, more or less, of a virus. So fortunately, this is a virus that produces very few deaths if you go to the number of people that actually infected. So the population at risk is considered to be around 40% of the population of the world, which is incredibly large. However, and this is the number of infections per year which are thought to take place. Around millions of people get infected. In most of the cases, it's a mild disease, it's a mild flu-like disease, But in many others, it actually becomes, with the dengue hemorrhagic fever or the dengue shock syndrome, then these are the cases that many of them then are fatal. Now, the reason for that is it's not very well understood why so little people, so few people do this. However, the numbers are these that I'm showing to you. Now, one of the characteristics of the virus, this is what I'm trying to show you here. Is the structure of the virus. So the virus is a flavivirus, and it's an envelope virus with a single-strand RNA genome. Now, the surface of the virus, you see, is a fairly round particle, and it's constituted by these blocks that you see here, and this is the only protein practically exposed on the surface of the virus, which is protein E, the protein of the envelope. There is another small protein M, which is just behind, and you don't see it here. So the whole thing, if you want to induce an immune response, which is based on antibodies neutralizing the virus, you need to target this protein. This has been the work of many people, and this is exactly what we have done. So it's a brief description on the protein. I will show you some more later on. Is that this protein that is exposed when it is synthesized, it is anchored to the membrane through two transmembrane domain, and then this is the part that is actually exposed and contains three different domains that are called D1, D2, and D3, with these colors that are represented here: the blue, the red, and the yellow color. Now the question is that in this protein you map the 4 different serotypes that exist of dengue virus. So there are 4 different viruses, and the different serotypes that have been characterized are dependent on the sequence of a protein E. And this is important because if you've been infected with one virus, most likely you will develop an immunity for that virus for the future, for many, many years. But if you are infected by a different serotype, then most likely you are not protected. And not only you are not protected, you are even in a worse situation, because the antibodies that actually recognize the virus but do not neutralize the virus are actually are increasing the infectivity of that virus. And this is because of this mechanism that has been described, by which viruses that are even a neutralizing antibody could promote the internalization of the virus through Fc gamma receptors, which are present in the primary cells that the virus infects, that are macrophages and dendritic cells. Therefore, if you have an antibody which may block, even a neutralizing antibody may block the entrance of the virus may still go into the cell through this mechanism. So if you have viruses that are antibodies that are cross-reactive and are not cross-neutralizing, and the best cross-neutralizing antibodies are not the only ones that do this, but the ones that block the exit of the virus from the endosomes. So these are the antibodies that are actually very protective. And in order to get these antibodies, you need to have antibodies which are directed against the right part of the protein E, and also of high affinity. Otherwise, it would get dissociated by the low pH of the endosome. And therefore, this is the challenge of the dengue virus field vaccine, is to try to find a way to obtain some of the characteristics that I am describing here. So as I said, you need neutralizing antibodies, and most of the time the neutralizing antibodies are directed against conformational epitopes. So you need these 2 characteristics, otherwise you're not going to go very far with any vaccine. So what do we need for a dengue vaccine? We need high antibody titers, and we need high neutralization titers. So and these, as I said, they must be against conformational antibodies, conformational epitope. And you need also high quality antibodies, which means relatively high affinity. And they have to be of low cross-reactivity unless they are cross-neutralizing. I mean with cross-reactivity antibodies that were elicited against one virus and react with another one but do not neutralize. It may neutralize serotype 1 but not serotype 2, and therefore you are worse off if you have antibodies against one virus. And therefore what you actually need is a vaccine that protects all 4 serotypes. Otherwise you are worse off if you do not have an immunity that covers all possible 4 serotypes. Now, this mechanism of enhancement of infection due to cross-reacting antibodies, what's called the antibody-dependent enhancement, ADE, and then you know you need high this but low ADE. And of course, you need long-lasting immunity, so they should be protected from many. So remember that most of the different serotypes are actually circulating in all parts of the world. So there are some regions in which one particular serotype is prevalent in one period, but then there are the others coming, and so they are more or less mixed. So in order to get that, what you need is to have the right antigen and the right way of delivery. Because if you don't have the right type of immunity, you're not going to get a protective vaccine. So this we've been centering exactly on that. So the main question that we were trying to do is to say which is the right antigen to do, and many people have already done this, identifying domain 3 of the protein. Let me show you the structure of the envelope protein. So the envelope protein is encoded in the virus in 2 large regions. One that in this region here encodes domain 3, and this part encodes domain 1 and 2. You see that they are encoded in mixed segments, domain 1 and 2, but they fall into these categories. This is domain 2, this is domain 1, and this is domain 3. This domain has been already identified by many people as one of the main targets of the neutralizing responses. Not all neutralizing responses go here, but it's a relevant one. And these are quite serotype specific. And therefore, this is a good candidate to use as the vaccine. But you have many cross-reacting antibodies that are actually directed against this part. So we initially thought it would be nice to avoid having in the antigen domain 1 and 2 and keep only domain 3. Now, what you are seeing here is the same molecule, but now this is parallel to the plane, to the surface of the virus. So what you are seeing here are the protein as it is organized on the surface, which are practically, this is the protein, this is the membrane, they are sitting on the membrane, forming this dimer that you see here. As it is also, and then these are the anchor, the transmembrane part that keep the protein on the surface, on the plane of the surface. So if this is the protein we want to use, then we say, well, why don't we take domain 3 and we put it in a construct and we see whether this is expressed in cells, in culture cells. This would be the protein expressed. This is a domain which resembles immunoglobulin domain, and this is more or less to a scale the size of the variable regions of an immunoglobulin. So you see that this is more or less the size of a single domain of an immunoglobulin. So an antibody recognizing this would have more or less this type of relative size. Now, if you take D3 and you try to express it in cells, and you can do any mammalian cells, test any mammalian cells, you do not get secretion. It's very badly expressed and very badly secreted. Now, if you, instead of that, you add this domain, and this is domain coming also from an immunoglobulin. This is the CH3, is a domain, is the last domain of the constant region of the immunoglobulin, which is a dimerizing domain. If you add this domain to domain 3, then you get secretion. What I'm showing you here is the extract and the supernatant of cells transfected with construct containing this or that structure. And what you see here is that you do get secretion only if you have the domain 3. So this is extremely important, as I said. So now, in order to get antigen available, you need to get D3 secreted. And with this strategy, you do get it. Of course, you have to engineer also adding a leader peptide in order to get it to the ER lumen. But that was not the only thing. The other important thing was to have enough amount of protein because some of them were expressed, although they were secreted, this is secreted material secreted in the supernatant, some of them are expressed at very low level. And the reason for that is the codon they use to express the process. We were using initially the codon coming from the cDNA of the viruses. So we now move into a codon optimized synthetic genes, and if you do that, then what you get is, upon codon optimization, all of them are very well expressed and very well secreted because of the CH3. And apart from that, So apart from the fact that you get good secretion of the material, now the antigen is also dimeric, and this is dimerized through the CH3 domain I mentioned before. And this is also an important aspect, because in order to activate B cells, you need to have polyvalent, at least bivalent or dimeric antigen. So monovalent antigen, monomeric proteins are very, very poor antigens. So you need something like that. This also helps the immunogenicity of our construct. Now, I'm saying this of the secretion because based on this, exactly based on that, we file a patent because we know now how to get D3 secreted, and this works for all 4 serotypes. So codon optimization and CA3 plus the engineering with the leader peptide, this is what actually is the base of a patent we have filed for a DNA vaccine for dengue virus. And I say that people do not do this very frequently. You may find several papers in which the whole ectodomain of the envelope, so which consists domain 1, 2, and domain 3, express, and particularly for serotype 2, in fact, hundreds of papers. And the titers you see are extremely, extremely low. And the reason is this is not secreted. If you take this and you try to express, and this doesn't depend on the cell, you can do it in any cell line, it doesn't get secreted. Now, if you do this with serotype 3 or 4, you do get secretion. So secretion is extremely important at the time of delivering this. Now, the reason why in the virus works, because there are other proteins of the virus, the PRN, that actually assist the folding and the assembly on the viral particle. And they do not get secreted because they do not get properly folded. We have here is a monoclonal antibody that actually is able to see whether domain 2 is properly folded or not. And while domain 3 and 4 you get in the supernatant, but also intracellular, but the supernatant, you get reactivity with this antibody. If you do with these 2, they do not. So domain 2, although the protein is there, these are all the controls. So that means that the domain 2 does not get properly folded in cells and therefore does not get secreted. This is an aspect to keep in mind. So what we actually did, we went, well, let's try our DNA immunization. So how do we immunize? Most common and frequent technology to do that is injecting DNA intramuscular. We do the gene gun immunization. The gene gun immunization delivers very small amount of DNA in particles that actually transfect directly intradermally, the keratinocytes. And therefore you start producing cells, you start producing the antigen in the intradermally. So this is the antigen we produce. We immunize groups of animals. This is the ELISA for all single animals and for the different pools of sera. And I'm not going to all the details. Just let me show you the titers we get are extremely, extremely high. This is 1 in 20,000. This is almost 1 in 45,000. This is almost 1 in 60,000. So this sera contains a concentration of antibody or in the order of 10 to 30 micrograms per milliliter of specific antibodies against the antigen. So these are extremely, extremely high levels. And many people get something like this also with protein, immunizing with protein. But there is a difference. We also show here that you can actually use the same material that we were using, or a surrogate of that, D3, to test our reactivity or the whole protein. Well, our antibodies recognize domain 3 in the concept of the full length protein. And you can see it here, cells infected with the different viruses and reacted with the different anti-D3 against serotype 1, 2, 3, or 4, and they are all detected as well as this. Intracellular, these are viruses. And then we did another ELISA in which we captured directly the virus itself. So do our sera see the infected viral particles? And the answer is yes. We have very good values detecting fully infected viral particles. Now, I was mentioning to you that an important aspect of this is to have the right antibodies. You want high titers, but you want antibodies directed against conformational epitopes. And is that the case in our? And the answer is yes. If you go and you go and do perform the ELISA using the native antigen or a denatured antigen. And you can do this just by boiling and loading on the plate of the ELISA exactly the same amount of protein, and we have the ways of controlling all that. What you see is an enormous drop in the ability of these antibodies to recognize this. So what our antibodies recognize is the native conformation. And this is the type of antibody that you want to have. You can also look at the quality of the antibody on an avidity index. You cannot do a true affinity because you have a serum. You don't have here individual one. And you do that by producing and doing the ELISA in normal conditions or washing at the very stringent conditions of 6 molar urea. So, and then you make the ratio. And if it is considered that you need antibodies above this level of 30 in order to get antibodies that have a acceptable affinity. And some of them are even much, much higher. Now, I wanted to mention that we got the highest titers we got against serotype 2. We also look how much cross-reactivity we have. We want to have, are they specific? And the answer is they are quite specific. Each one of them reacts against itself and very little against the other serotypes, with the exception of serotype 3. So what you have here is each one of the serotype and all 4 different proteins, and the anti-serotype 3 reacts with serotype 1, 2, and 4 at different levels, but this is a bit cross-reactive, while the others are not. You do not have. You have very little cross-reactivity. Now, obviously, the most important thing is, but these antibodies, are they neutralizing? I mean, do they block virus infection? And this is what we did here. This is an assay, for instance, for the serotype 2. And again, we did every single one, and this is what you have for the pool of sera. So we have titers. These are neutralization titers of 1 in 300. And these are extremely, extremely high. Many people show for dengue vaccines and DNA vaccines, and they are right to 1 in 50, to 1 in 60. In the best cases, and others, and sometimes for the ones which are more immunogenic in mice, because we are in mice. And this is serotype 2. You see in serotype 2 we have 1 in 1,600. And these other 2 we have 300. The one that doesn't work very well is serotype 4, and we are working on that, trying to solve this situation, because it's already pretty good. We are close to 100, but it's not as good as we would like it to be. Now, if you go to look, but are these the cross-neutralization? Before I showed you that there was very little cross-reaction based on an ELISA. These are cross-neutralization assays. So you take against sera, elicit against one serotype, and we check against all the others. And you see that each one, this one, protects only serotype 2, doesn't do absolutely anything to the others. This one does exactly the same, very, very little against all the others. While serotype 3, that actually was cross-reactive, is also somehow cross-protective, cross-neutralizing, particularly against serotype 2 and a bit to serotype 4. So this could also be one strategy to try to get a broader coverage, because if you have cross-reactivity, that it is cross-neutralizing, then you are on the right way. And again, the serotype 4, as I said, is the one that is the lower one. But nevertheless, if you compare with what is published, this is quite acceptable value. All these are obviously much, much higher. Now, I'm not going to detail into that. We just wanted to compare whether what happens if we now take the whole ectodomain, domain 1, 2, and 3, or domain 3 only, and we compare the immune responses. What happened with that? Well, what actually happens is that most of the immune response, if you give the whole envelope protein, is against domain 1 and 2, the one which is more cross-reactive and less protective, which you can see here. You see here that the reactivity you get against the domain 1 and 2 only, or the whole envelope, is more or less the same, while what you get against domain 3 is very little. So if you want to use if you want to induce an anti-domain 3 response, you need to use domain 3 only and not the whole protein. And this is the neutralization we get. This is the neutralization we get and this is tested also in a large number of animals, sera against domain 3 of the same serotype or the whole protein. You have this value which is almost 3 times lower when you use the whole protein. And the reason for that is because you have plenty of antibodies that are directed against D1 and D2, which are less neutralizing than the ones against directed 3. So it's not that there are not, there are some of them, and recently have been described some of them, which are very efficient, but are very rare, are not the more frequent ones. So this is again to show you that I already mentioned codon optimization was an important aspect because you increase a lot the amount of protein you get. So you need to do 2 or 3 different things in order to improve the quality of the antigens. Now, this is the last piece that we wanted to have an idea. So far, all the data I showed to you was we were using 4 different constructs in 4 different group of animals vaccinated with construct 1, 2, 3, or 4, and asking what happened with each one. We wanted to understand each one by one what happened. But in the end, we need a tetravalent vaccine. We need a combination that provides protection to all 4, neutralization to all 4 viruses. And therefore, this is the very first test we did, and we have others now going on, trying to obtain a formulation that actually improves or gets an acceptable level for all 4 of these ones. So, and there are many variables that have to be taken into consideration. What I'm showing you here is the reactivity of these sera comparing the monovalent and the tetravalent vaccine, and you see that in some cases they are very similar. In others, what we got against the tetravalent is lower than what we got against the monovalent. Particularly the titers we get were somehow reduced, but nevertheless were acceptable. These are the titers of the tetravalent compared with the titers of the monovalent. And look at the case of serotype 2. We have an enormous drop in the antibodies induced in the tetravalent when 2 is present when we do it alone. So this is already an important thing to learn, because the vaccine does not, and this was a mix, so we were doing, in this case, we were mixing the constructs. But there are other strategies that you can use. You can, at the moment of the vaccination, you can vaccinate domain serotype 2 on one part and the others on a different one, because apparently there is here a preferential, a more compromised or less efficient activation of anti-serotype 2 antibodies when you have all of them mixed. Some others are very, very much the same, as I mentioned before, for serotype 1. But nevertheless, the quality of the antibodies, this is again the avidity index, the quality of them are more or less the same as the monovalent. So from this point of view, we are quite safe. We need probably to get a better formulation to increase protection, and particular for the neutralization. So the neutralization titers, these were the neutralization titers for the monovalent, the tetravalents. You can see the curves here. And we get, in 3 cases, more than 100, which is already acceptable, but of course we would like to have more than that. And this is the one which is more compromised. So again, we went down here from around 70 to 25, and this has to be improved in order to have so serotype 4 is creating somehow of problem. But nevertheless, this is, as I said, the very first attempt of a tetravalent DNA vaccine with this, which is extremely promising for all these. Particularly when you compare what there is around. And also because of the more classical, I'm finishing 1 minute, Mauro. The more classical vaccines that have been tested, many of them have actually failed. And the reason for that, I think, is because you cannot continue doing classical strategies of vaccines for problems, complicated problems like dengue. One has to be innovative and using strategies like this or others that could actually make the breakthrough to get the right protection. So just to finish, let me tell you what we have done. I think there are the 3 main elements that actually make our vaccine promising, and we have now a company that is interested in pursuing with this and further develop, because there are a number of things that have to be yet optimized. The first, that we use a DNA vaccine. So this makes an enormous difference. We also have experience. I didn't show you the data, but if you want to use, instead of doing with the same cocktail, you produce the protein ex vivo and then you vaccinate, you don't get that number of conformational antibodies. You do get them, but you get plenty of other responses which are not useful at all. So they are more cross-reactive, and you get plenty of antibodies that are directed against epitopes that are not conformational. You don't want them, particularly in the case of dengue. This is more important. In other cases, you may not care about them, but in the case of very simple. So DNA-based, I think, is the way to go. The second thing is, of course, we had to engineer that, and in order to do that, we have to introduce this modification. And fusing to see the domain CH3 of the immunoglobulin was the key thing that actually allows the breakthrough. If we didn't have that, the whole thing would not have been possible. The whole thing was possible because of this that allows secretion and also help with the dimerization. And the third important point, which I didn't show you any data, but we do have data also that, is the way that we do the delivery of the DNA. We do gene gun and not intramuscular. We have data comparing both. You can use 50 micrograms injecting in the muscle and compare with the neutralization obtained with 1 microgram of gene gun, and the difference is like this. So it's enormous. You get much more and better responses if you do gene gun because it is intradermal and not intramuscular than if you do it in other ways. And we are also trying to obtain and to follow other alternative ways of delivering like viruses and also others. So, well, I stop here. Let's just let me thank the two main people that have actually contributed to this project, Monica Pojanella and Jose Luis Campos, and Sara Marquez joined a few months ago, the group working on this, and Marco Pestaño, a collaborator that also helped on the project. Thank you.